of the Society of Graduate Students at Western University. I'm your host, Anna Moyer.
And I'm your co-host, Elizabeth Mohler.
And we are here with Carlie Muir today. Thank you for being here.
Thank you for having me.
And I think we would like to start off today by just asking you, maybe some opening questions about what salmon spawning is and how did you get into this area of research?
Right. So salmon, I would say, are one of the charismatic animals that we have here in Canada. And their spawning is one of the things that's so interesting about them. I think there's even a Magic School Bus episode about it. And so what's really interesting is that salmon are spawned into these rivers and they develop there for a couple of years before they migrate out to sea. And then when they come back to spawn, they're returning to the same natal rivers in which they were born. And so that's something that I think has always fascinated people. And so sort of what I'm looking at is sort of what goes on in that developmental period and sort of how did the conditions that are experienced during that time sort of prepare them for life and the conditions that they'll experience down the road.
Sorry, could you maybe, that was great. Could you give us just a little bit more context on, what spawning is for those who might not be familiar with it and how it happens?
Sure. So salmon spawning is basically the process by which females are coming into these freshwater streams and laying eggs, and then the males are coming and fertilizing those eggs. And so what's really interesting about some species of salmonids, so your Pacific salmon like Chinook and sockeye, is that they only make this spawning migration once. So they only breed once and then they die. Whereas Atlantic salmon, which is the species that I focus on, they make multiple spawning migrations. So they're sort of going back and forth multiple years on these spawning migrations where they return to these natal streams, the females are depositing eggs and the males are fertilizing them, and then they're going back out to the ocean. And so this is called an anadromous lifestyle. And what that means is that their life history is divided between both freshwater and saltwater, and they involve these really aerobically demanding migrations to go back and forth. And that's really an evolutionary bottleneck for them because so much of their fitness is tied up in whether or not they can make these really labor-intensive migrations upriver.
Wow. So in terms of your research, how does this play into what you're actually studying?
Right. So what we've found as people have started sort of paying closer attention to the thermal tolerances of animals is that salmon are really locally adapted to the thermal conditions of these natal streams. So the conditions that their ancestors experienced on their upriver migration has put the selection pressure on future generations such that they perform their best at temperatures that were experienced by their ancestors on these migration routes. And so with climate change, what we're finding is that so many of these populations are narrowly adapted to the past temperature regimes, such that their performance is optimized near average summer temperatures, and they sort of die around peak summer temperatures, the current ones. So what we're finding as climate change increases the frequency of heat waves in these record heat events, sort of like what we were having a couple weeks ago, it's really causing these salmon to die off. We're seeing these mass die-offs along their migration routes and in the juveniles that are living in these streams because these streams are so thermally unstable. And so the issue is the rates at which the climate is warming doesn't really allow for enough time for these populations to evolve to the new thermal regimes. So that's sort of where my work comes in, is I wanted to look at sort of the capacity for plasticity. So how one genotype can produce multiple phenotypes within one individual's life history. So how did the conditions that an embryo is experiencing during development going to change the phenotype, so change sort of the traits it expresses later in life. So if an embryo is developing in a river temperature that's a few degrees warmer than what the mother or previous generations experienced, will that then alter their physiology going forward so that they're able to withstand higher temperatures? Or are they sort of locked in to sort of what they evolved to withstand.
And you used a couple of terms. I'm just wondering, could you sort of clarify genotype and phenotype and what those terms mean?
Right. So your genotype is essentially what your DNA has encoded for you, what it sets out and predetermines. And a phenotype is sort of how that trait presents. And so one genotype can produce multiple phenotypes based on the environmental conditions. And so that's what we call plasticity, is sort of the ability for one genotype to produce multiple phenotypes. So you can think of the genotype as sort of the code as it's written, and the phenotype as the product that's sort of presented.
Okay, wonderful. So what are your findings so far in terms of the phenotype actually expressing itself?
Right. So what I did in my PhD was I took siblings of salmon from one spawning and I divided them into 2 rearing treatments. So one set we reared sort of under the current environmental conditions, so current temperatures. And the other set, we reared them at 4 degrees higher throughout development to sort of mimic the projected increase in temperature by the year 2100. And so by taking ones that had sort of the same genetic background and rearing them in these different environmental conditions, we would be able to infer that any differences were due to the environmental conditions that they experienced during development. And what we found was that when we raised these salmon at temperatures 4 degrees higher, they were actually able to maintain their function to higher temperatures. So their optimal temperature, which is physiologically, it's where their scope is maximized, just where their performance is maximized. We found that kind of tracked the increase in environmental temperature 100%. So their optimal temperature shifted 4 degrees along with that shift in developmental temperature, but their upper lethal limit only shifted 2 degrees. So this is still positive. There is a degree of plasticity in that, but it does suggest that perhaps that upper lethal limit that fish have is a bit more rigid. And that's concerning given sort of the climate change projections that we're facing.
And why do you think that upper lethal limit might be so rigid?
I'm not sure if it's sort of a lack of genetic variation in the upper lethal limit, or if there's been a bottleneck and so they've sort of maxed out their physiological capacity. Because something that you hear sometimes in physiology is that we have plastic floors and concrete ceilings. So there's more wiggle room in sort of how you function at sublethal temperatures and less so in that upper lethal limit. But it's also not lost because in addition to sort of shifting their physiology and how well they can actually withstand these high temperatures, salmon can also shift the timing of those migrations that we were talking about. And that's not really what I study, but it's something that we're starting to see is that instead of facing these warmer temperatures, salmon are migrating later in the year, say when it's to avoid sort of the warmer summer temperatures and things like that. Although there are also caveats to that. There are carryover effects that can have. But yeah, that's sort of one option that we may see salmon populations go down as the climate warms.
Now, this question might be a little bit outside of your area, but I do know that there are some projects on revitalizing rivers by putting tree roots back into the riverbeds to help kind of deepen the rivers and control temperature. Have you seen anything on that? And does it actually positively affect salmon populations?
So interestingly enough, part of my PhD work was done in collaboration with the Lake Ontario Atlantic Salmon Restoration Project, which is basically a push to bring Atlantic salmon back to Lake Ontario. And so part of that project is how can we restore sort of these natal streams to make them more conducive to juvenile salmon survival. And so I don't know specifically about planting trees, but yeah, if you think about it, if you can offer sort of thermal refuges where fish can go and sort of find a cooler pocket, that's something that physiologically would be super beneficial for them. And so just to tie it back to the work that I was doing, part of what we were looking at was we're finding that when you stock, if you rear salmon in hatcheries, which is something that the MNR does, and then you stock them out into streams, one of sort of the limiting factors is that the salmon, once they get the juvenile salmon, once they're in the streams, they can't withstand the temperature. So their thermal tolerance from living in a hatchery hasn't really prepared them for being in these thermally unstable streams. So that's part of why we want to see, can we raise the temperatures that we rear these fish at in the hatchery before we release them? And will that sort of better prepare them for life in these streams? So pairing sort of how we rear the fish as well as making these habitats more suitable for them, because there's been so much habitat destruction sort of before we stop to think about, well, we may actually want to have these fish populations around. So.
And what have you found so far? Have you found that, when you've raised the temperature, the fish are able to sort of withstand going out into the big wide world, as it were?
So right now we've just sort of tested in the lab to say, yes, it looks like if we raise them at these higher temperatures, they're going to be able to withstand higher temperatures, but we haven't taken that step yet to sort of tag them and send them out and see how well they survive. So that'll sort of be the next step if the MNR and the Ontario Federation of Anglers and Hunters decide to sort of take our recommendation.
And I was wondering, is there any difference in how the fish react to, for example, a short heat wave versus a long scale temperature rise?
Yeah, that's a really good question. And so that's something that my work has also sort of tried to tease apart. And it brings back what we were talking about, those plastic floors and concrete ceilings, because the longer term average increase in temperature, it's really going to matter sort of. Sorry, I guess I should take a step back and sort of explain what's going on physiologically with fish when we're at these higher temperatures. So as ectotherms, their internal physiology is really tightly linked to external temperature. And so they don't have a constant body temperature the way mammals do. So when the external temperature increases, that increases the reaction rates of all of their biochemical reactions in their metabolism. And so our metabolism is aerobic, it requires oxygen. And so as these reaction rates increases, the oxygen demands increase at the tissues. And so in fish, it's thought that they're limited sort of by the cardiovascular system's ability to deliver oxygen to tissues and meet these higher oxygen demands. And so when we think about sort of a higher average temperature versus a sort of these spikes in temperatures, we're sort of thinking about it in two different ways. So how much does sort of coasting at a higher average temperature take away from your energy that you have available for things like growth and reproduction? So it's sort of slowly siphoning away your aerobic capacity from other things that would contribute to your fitness. You can imagine that if a fish sort of is always at this higher temperature that's above its optimal temperature, it's going to have less energy to invest in eggs, and it's going to have a lower reproductive output. Whereas their upper tolerance, that's sort of an acute warming response. And so how well are they able to sort of survive a one-off spike in temperature? And that's kind of what the experiments I did was looking at, was how well can they respond to these acute spikes in temperature? And that's what we were looking at in terms of the 2-degree increase. So they can sort of survive to 2 degrees higher, whereas their optimum is 4 degrees higher. So that suggests that they're almost tracking the temperature better in terms of their optimal temperature. So sort of the average increases, but when it comes to that upper lethal limit, there's less flexibility like we were talking about.
So this might be outside the scope of your research. You talked a little bit about, the upper temperature and withstanding heat. Have you sort of gone the other way to see how much lower the temperature can go where the fish will withstand?
So I haven't done that, but others certainly have, especially in a paper, I believe it's out of the Farrell lab, Matt Gilbert's work, and they were sort of looking at Arctic char, and they sort of did similar work that I did, but in both directions. They raised them as high as they could go and took them sort of as low as they can go. And yeah, obviously when you're increasing the temperature, we see that their heart rates are increasing. up until a point where they can no longer do so and they're no longer to meet those increased tissue oxygen demands. And it's sort of the opposite. When you cool them down, the heart rate slows and they're no longer able to sort of move blood throughout the body because the heart is pumping so slowly. But then again, their metabolic needs are so reduced at those lower temperatures.
And this might be not the best question, but I was kind of curious when you mentioned that there wasn't a lot of genetic variation, and this may be a reason why fish are less able to adapt. Is this in part due to things like overfishing or the fact that salmon are not able to spawn in areas like Lake Ontario that they used to be?
So I'm not sure. So there's not sufficient genetic variation. That's not to say they have less genetic variation than sort of other species. But in addition to the lack of variation, there's just not sufficient generation time for adaptation to act. I'm not sure how much fishing practices impact the genetic variation in terms of thermal tolerance, but that's an interesting question.
Do you study like a specific type of salmon? Because I mean, there are lots of different types. So could you tell us about that?
Yeah. So I study Atlantic salmon. Again, the reason we're interested in Atlantic salmon is because we have this partnership with the Atlantic Salmon Restoration Project in Lake Ontario. And again, Atlantic salmon, I think they get less attention here in Canada because people find, sockeyes a little more charismatic. Those are the ones that are bright red when they spawn. But the Atlantic salmon are neat because they have more than one spawning migration. They're Iteroparis, I believe. I'm going to get roasted by fellow salmon biologists if I'm mixing up Semmelparis and Iteroparis, but I believe Semmelparis is when they only have one migration, spawning migration, and Iteroparis is when they have multiple, or they have the capacity to do multiple. But that's why I think that Atlantic salmon are so interesting.
So what kind of triggered you to begin studying salmon, and specifically Atlantic salmon?
Oh gosh. I mean, how did I find myself here? Such a winding road. I actually started out in research studying cell biology. I was looking at kidney epithelial cells from a frog and ECM remodeling. And that's what I did my honors project on. It was a developmental cell migration heavy project. But then I just sort of craved, I had always grown up loving science and loving nature. When I was a kid, my dad likes to joke that I would tell people I was going to be Jane Goodall when I grew up. And so I craved something that had more of like a conservation application. And so I sort of made a space for myself where I kept working with the supervisor from my undergraduate project, who was a developmental cell biologist. And we partnered with a fish biologist who was doing this conservation work, and sort of thought, how can we create a project that sort of brings my developmental molecular toolkit to sort of this salmon conservation. And that's where we decided to start looking at developmental plasticity, specifically temperature dependent plasticity.
And where do you think you might go from here? Do you have any plans for future studies, or?
I'm actually looking into policy opportunities right now. It wasn't my initial plan. I think I went into this expecting that I would continue on and do a postdoc, but just sort of the last couple years, I've started thinking more and more about science policy and how we need more people, more scientists working in government and sort of fighting for evidence-based decision-making in our policies. So that's where I think I'm headed now.
Fantastic. Are there any good examples of policies that you might be working on in the future?
I'm not sure, because I'm just sort of starting to have my interviews and stuff, but I would love to. So Canada's actually working on its first aquaculture act. Canada, and that's something that I would be really interested in working on if possible, but I'm really open to anything sort of climate change, fisheries. That's sort of my bag.
All right, so on a slightly different note, you mentioned that the heart function is a very specific issue with the salmon and their ability to kind of generate oxygen or produce, live. And so I was wondering what the morphological or functional traits were that would allow a salmon's heart to kind of evolve to deal with these higher temperatures.
Yeah, that's a really great question. So there have been a bunch of studies in this area, and what a few of them have found is that the fish that are able to survive to higher temperatures or maintain their heart function to higher temperatures are able to achieve higher peak heart rates. And then there are other studies where fish that have a bigger heart, so that means they're moving a bigger volume of blood with each beat, are also able to maintain aerobic function to higher temperatures. And so heart rate and stroke volume, which is what you would get from a bigger heart, are sort of the two components of cardiac output. So what this suggests in general is that fish that have can produce a higher cardiac output can survive to higher temperatures. And so that's what I went into my project expecting to see, but I actually did not. But I did see some differences in morphology. So what I found was a thicker compact myocardium in the hearts of the fish that were surviving to higher temperatures. And so the fish heart is different than a mammalian heart because it's only two chambers. And the ventricle is the chamber that's really doing a lot of the legwork. So the salmon ventricle, it's this pyramidal shape, and it has two layers. It has an inner spongy layer that gets its oxygen from the returning blood in the lumen. And then it has this thick, powerful compact myocardium that's receiving a coronary blood supply. So it's sort of getting the freshly oxygenated blood. And so what I found was that the fish that had a thicker compact myocardium were able to survive to higher temperatures and maintain their heart function to higher temperatures. And so the thinking here is that perhaps the hearts themselves are just better oxygenated, and that allows them to continue functioning under these high temperature conditions and continue beating, sending oxygenated blood to the body tissues. So this is in line with sort of what some people have shown, in that others have shown when you sort of cut the coronary blood supply to the compact myocardium, that reduces thermal tolerance. And so, yeah, my project sort of suggests that perhaps the functional trait in these fish is lending to a preservation of heart function rather than a greater overall cardiac capacity. So instead of moving more blood per unit time, it's just that the heart is able to withstand higher temperatures and it's continuing to function to higher temperatures, if that makes sense.
Yeah,
so
no.
It's not the pound for pound function. It's just that they're able to withstand higher temperatures because they're better oxygenated.
Yeah, that makes a lot of sense. I just wondered if Carlie, it does look like we're thinking about wrapping things up, but wonder if you want to share any social media with us or any way folks can get in touch with you?
Sure. I'm on Twitter. My Twitter handle is just my name. So it's at Carlie Muir, which I'm assuming will be in the show notes here. You'll be able to get the spelling. Yeah.
Well, thank you so much. This has been GradCast, the official radio show and podcast of the Society of Graduate Students at Western University. I've been your host, Anna Moyer, and my co-host was Elizabeth Mohler. We've been speaking with Carlie Muir, and this episode was produced by Ariel Frame. If you'd like to be involved with the show or get in contact with us, email us at gradcastradio@gmail.com. You can follow us on Instagram, Facebook, or Twitter at GradCastRadio. To listen to us, we're on Radio Western 94.9 FM. You can also find all of our episodes on our website at gradcast.ca or on podcast apps like Podbean, iTunes, and Spotify. Alternatively, select podcasts have been uploaded to YouTube at GradCast Radio. Thank you for listening and have a great night.